Axel. All right. So Axel asked me just 2 days ago at Mark's defense, so I only started thinking what I would say this morning. So you'll have to. It's very informal. You have to excuse that. I guess what I wanted to talk about. No, I want to start with a take-home message, just in case I've really made a mess of the talk. Most of the way that people in biology have been working on this question of early evolution is looking at it from a historical point of view, and that means trying to see backwards in time by comparing modern organisms. How far back can we see similarities, and can we then recreate the states of earlier organisms or earlier systems by this approach? I guess the take-home message I want to put across is that now that we've got much more data, particularly from genome sequencing projects and from non-standard organisms that we don't tend to talk about when people write textbooks about sort of how cellular systems operate. What we're seeing is so many exceptions that the rules are pretty much vanishing. And probably what we're also seeing is that much of the historical trace just doesn't actually exist. So it sort of looked like it existed, but it's disappeared. And so maybe from your point of view as non-biologists, perhaps the really interesting part of that is that I think we're starting to move towards trying to look at biology in terms of systems and how do systems operate, and can we understand the conditions under which certain types of phenomena that we've sometimes thought were associated with being old or being recent can emerge and re-emerge by perturbations to those systems. So I'll try and make some sense of that as I go through. We'll see how we go. Okay, so I guess this statement says it all. It's a rather provocative statement from a review by Chuck Kurland, who says modern cells present no signs of a putative prebiotic RNA world. And I think that would have been a heretical thing to say a little while ago, but that's probably true. So just to give you some background on what that is, I guess everybody's aware DNA is the genetic material, and we have this process by which the information in DNA is expressed, and the common product we get from that is proteins. We have a working copy as the message, a protein is produced. That protein proteins are required to replicate that DNA, so there's an interdependence between the two. In the 60s, when people were looking at this, shortly after the structure of DNA had been established and the genetic code was being worked out, there was a lot of speculation about this classic chicken and egg problem. If DNA carries the information from which proteins can be synthesized, and proteins are required to make DNA, you have this chicken and egg problem of which came first. And the model that was proposed in the 60s was that RNA could be in the genetic material and could also be in an enzyme. And it was actually known already in the 50s that RNA can be a genetic material. It's found in things like tobacco mosaic virus, so that's one good thing that the tobacco industry has given us. So we know that RNAs are present in all sorts of viruses, things like the influenza virus that some of you may have been unfortunate enough to have, SARS virus, HIV has an RNA stage, and so on. And in the 80s, RNA enzymes were discovered, so that sort of suggested that in principle this type of a model could operate. So that would look something like this, perhaps. So instead of having DNA and proteins, we have an RNA genome with working copies being produced, perhaps processed, and you have functional RNAs that will be required to make that RNA genome.
So there is no protein involved in the previous slide.
Well, people have shown chemically this can happen.
Yeah.
But not well enough that you could copy a piece of RNA.
Does it happen with a host?
So what you're in a virus? No, you need proteins,
yes.
Or the host protein proteins.
Yes.
So there are no self-supporting RNA-based systems that are being produced.
So to what extent is then this sketch accurate?
It's accurate in terms of we know that the chemistry is possible, but nobody's perhaps found an enzyme that's quite capable of doing this.
Yeah.
So it's it's sort of, in principle, it's possible, not demonstrated.
Yes, yes.
In the previous slide, you talked about DNA going over to exactly right. Is it possible for DNA to replicate itself outside it, like for example, in a PCR device? And there do you go through the same cycle or you don't?
Right. No. So DNA, if you run a PCR cycle, you're adding these guys in, and they're copying that. But the information that they carry is nothing related to this. So they're doing this half of that reaction there. Proteins are just taking a template and making more copies of it. It does turn out that it transpires that DNA can, under some, there are some catalytic properties to DNA, so it can do chemical reactions, sometimes quite well, but that's still a long way from
between proteins and DNA, you have code. Code, of course.
Yes.
It's not just copying.
No.
Or
That's
PCR is just an amplification of.
Yes. Exactly.
Of dimers, of a double helix.
Well, you don't need to have information based on proteins in the template, obviously. But if you want a self-supporting system, then there has to be some way of getting that information into that, and that requires a genetic code, and there's a very complex machinery for that. Absolutely.
Yes.
So, this is sort of a contrived chicken and egg problem, if you like. It hides most of the complexity.
Are there any RNA enzymes in living cells found?
Yes, yep, we have. Yep, there are RNA enzymes in everybody's cells, and in fact ubiquitous to all life. So there are some good examples of these, but they're probably also very ancient.
What is, for example, the best?
I think I have a slide with a couple of things that we think of. So I guess what we can say is, assuming this hypothesis is reasonable, what it implies is that there are two transitions that have occurred. One from RNA to DNA. When we're now in modern cells, DNA is the genetic material, and then one from RNA as the catalyst carrying out the reactions to proteins performing those reactions. So the standard picture then is that if we trace backwards from modern cells, we can go to these earlier states. So the common ancestor of all life, we often shorten to LUCA, which is just short for last universal common ancestor. So that just means the great grandmother of all cellular life forms. At some point prior to that, we had the emergence of things like genetically encoded proteins and probably DNA, and that earlier than that, it's an RNA world. So I guess the key questions then is, what is the evidence for these two transitions, and are there relics, I guess living fossils in cells that show that this earlier stage existed? So the basic model or assumption is that under evolution with natural selection, we would expect for transitions to occur in the following directions. So state 1 goes to state 2, and state 2 is better than state 1. So in that previous slide here, what we're saying is that DNA would have to be better at RNA at this job, and proteins would have overall to be better than RNA at that job. Better as in if there were two cells, one which carried out the process using the tools of state 1 versus one carried the tools for state 2, the state 2 cells would predominate, and state 1 would go extinct.
But in this case, you are talking about a system which actually requires both. I mean, any protein requires still RNA to operate.
Yeah, it's not black and white. I'll try and get to that.
Yes.
Okay, so it turns out there are actually, we tend to think of there being one way to make proteins in cells, and there are actually several ways that this happens. Proteins can be made in this way here, which is using the genetic code, so there is information stored on these working copies of genetic information, and they will be decoded by a machine called the ribosome, and the ribosome is doing two things. It's reading the information in the message, so it's reading the genetic code, and it's also translating that and performing this protein synthesis reaction to make these building blocks here. Join the building blocks together to make a protein. And the key thing there is what's been discovered is that both of these processes are performed by the RNA core. So even though this is a machine made predominantly out of proteins, required to make proteins, all the stuff that's going on there in terms of the reactions is being done by the RNA. And so what that sort of suggests, that's what you may expect for a transition from an RNA to a protein state, is that the machinery to do that is made from RNA. However, we also find other cases like this. This is a terrible picture, but I'll walk you through it. What this is showing is a modular system, an alternative modular system for making proteins, and we call this non-ribosomal protein or peptide synthesis. Basically, we have a number of different, each one of these blocks here is a protein module, or actually a domain, and a couple of 2 or 3 domains may make up a module. And each one of those modules has a role in making a product. So what happens is there's a module here, A, which selects the appropriate substrate. This here will hang on to the existing made product. So you've got the thing you want to join onto your existing big product. This condensation domain does the reaction, and then select another substrate. You're holding on to the piece that you've already made. You may add some bells and whistles with these types of things and stick it all together. And at the very end, you've got a release domain here that allows the product to fall off.
So this is almost like an assembly line in a factory.
It is exactly like that.
Is it like, I mean, what is this? This is an RNA or?
No, this is a protein. So what these are is, each one of these is quite a large protein module. So you have a whole series of these, and they might make a big...
so it's a macromolecule made of protein
large,
pieces.
yes, yes. So you have this very, very large, extremely large enzyme that has this assembly line feature of being made up of these smaller modules. And each of those modules is making a little piece of peptide, and at the end you'll get a bigger peptide product. So things like antibiotics get made by this type of process.
But each colored block is another protein already.
Well, it's a domain of a protein, so it's a piece of a protein that can do a specific job.
And how long is it?
These things they can be really, really large. So each one of these, this is about 5, you can see this is 500 amino acids, 100 here, you get another 500 there. So you get, just for one module there, you have quite a large protein already.
Okay.
So
is any folding structure in that business important already?
I suspect so. I don't know about the details of the folding. But the key thing about this is proteins making proteins rather than RNA making proteins. So that's important, especially when we think about that inflammatory statement at the beginning that there's no evidence for an RNA world. The point here is that this is proof of principle that protein can make protein. You can have a simple modular system where you're repeating the same units and making slightly different components which you join together to make a large complex.
There's this so-called Gadieri reaction. Is that part of such a thing?
I don't know what that is.
It is replicating, it's a specific replication of proteins, I thought.
Okay, this might be
Gadieri.
different.
Could be.
Right, no, okay, I'll look that up. So what these are then are what we call large, mega enzymes. They're very, very large. And of course the problem from the perspective of an origin of life is that the information input is far, far greater than the output. You might have one of these mega enzymes is tens of thousands of amino acids long, but the product might be 20 or 30. So you've got several orders of magnitude difference between the information input and the output. So
in the natural world, what makes these mega enzymes?
what makes it? Well, that's ironically this is made through this. So the information, sorry, not through this. It's made by one of these, a ribosome. So the information is stored in the genome. You can make them, but of course the only difference is that rather than the proteins being the information being stored here to make these small products, the information is effectively stored in the information here. So you could say ultimately it's stored in a genetic information, but it requires this to be produced to then go and produce a subsequent molecule. So I guess that the bottom line is that one we're seeing in biology that there are other ways to do this than the one that we thought there was only one way to do this. We're now seeing that there are other ways. It's not really direct evidence for an alternative transition in life, probably because of this type of problem, but this is something that perhaps from a chemical point of view can be examined to see whether you could make a much more minimal system which is smaller and perhaps cut down the discrepancy here. There's no reason why that couldn't happen in principle. There's also two ways to make DNA. We tend to think of it as having happened like this. Basically, in modern cells, you have building blocks for RNA, so there are 4 bases that constitute the information carrying part of an RNA molecule, and there are 4 bases in DNA. So the only difference between RNA and DNA in terms of the information carrying is that you have, instead of a U here, you have a T in DNA. And if we look at the pathways required to synthesize the DNA building blocks, your starting point are the RNA building blocks that carry out this reaction called ribonucleotide reduction, but you end up, because one of the inputs is a U here, you end up with a deoxy U. So that sort of meant that we thought that quite probably there was a 2-step transition, one to a uracil DNA and then to modern DNA with thymine instead. So this happens as a separate step.
So the RNA and the DNA, they are different with respect to the sugar in the backbone?
The base in the backbone. The sugar is, in the RNA the sugar is different as well. So I think I even have a picture of that. Yes. So the real difference, other than the base, is it's actually the sugar itself is almost the same. It's just that one of them is in the deoxy state and the other is kind of hydroxyl here at this position. And it turns out this actually gives the reactivity to natural RNAs. This is why they can carry out reactions, whereas this hydrogen is obviously going to be inert. But the reason why that reaction is significant is that process of the first step, changing it from a building block from an RNA building block to a DNA building block, requires this really horrible reaction here, which I won't go into other than to say that it's basically, it requires stable protein radicals. RNA can't do this. If you get RNA in here, this will just cleave the backbone. So there's no way that this reaction is amenable to RNAs. So
what cleaves the backbone?
Just the production of one of these radicals.
Uh-huh.
So people use, for example, when they're trying to determine the structure of RNAs, one of the techniques has been to probe it with radicals, and then it'll just indiscriminately cleave. So you can say, based on the distance from the source of the radical and the position of cleavage, you can start generating information about the structure.
Mm-hmm.
So yeah, RNAs just simply can't handle this type of chemistry. So as a result, that places DNA after proteins. You need to have got proteins before you could control this type of reaction. And so from that point of view, it seems that this is quite an old thing. So we know that this enzyme that carries out this reaction, ribonucleotide reductase, this is universal. All cells do it. It's the only way to make DNA in modern cells. As I said, it's too chemically complex for RNA, but there's a problem there, and this is one of the things I was mentioning. Evolutionary history is sort of erased because it turns out that we can't really trace it back to this Lucas state here, simply because there's ongoing transfers of the genes. So the genes that code these guys tend to jump around between the domains, which means we can't build a tree of them and place them back here. So even though they're probably there, another process is confounding that evolutionary signal. So I said there's 2 ways of doing it. This is the other way. This was actually the way that people originally thought DNA building blocks would be made. And this is, so down here at the very bottom we have our deoxyribonucleotides. So sorry, this is not a very good picture in the sense of understanding it. So normally what happens is to recover deoxyribonucleotides, they get shunted down this way in normal cells, and they get pushed into a process called glycolysis, which is just the way in which cells break down sugars. So this is the natural direction here, and the reason it's drawn the other way is that there have been efforts in an experimental group in Japan to try and push this in the other direction. For this, not because they're interested in origin of life studies, but doing it because they want to make these things are going to run out. I guess everybody's aware that there's lots of genome sequencing projects at the moment. These are becoming a commodity that are risking getting scarce soon because there's not enough of the raw materials for you to do your experiments. So they're trying to look at industrial processes to synthesize these, which means what they're trying to do is push cells to, in addition to making the DNA building blocks they need for replicating their DNA and for reproduction, produce an additional source by shunting these intermediates that you find normally in glycolysis through this pathway here to make more DNA. So it turns out that this works. This is a reaction that's found in most, probably all, I haven't actually looked, I should see that, cellular systems. So it's a widely distributed, simple, chemically simple system. So chemically speaking, there's no reason why RNA couldn't do that reaction. It's just that it's not used normally in biological systems. It's the reverse reaction. The reverse reaction is just not available. So what's important there is that could make it possible that DNA predates ribosomes. If that was actually used and it's no longer being used, then we can't say that DNA had to necessarily needed to come after proteins because there's a simpler way of making it. Okay, so that's kind of, maybe that gives you a sense that there's reasons to doubt that our textbook picture is as black and white as textbooks like to have us believe. And the second question then is, are there relics of this? Well, perhaps the one that everybody picks up is this one here, the ribosome, where I said, of course, it has an RNA core, and that RNA core is what was used to build the tree of life. So because every cellular system uses this, you can compare the sequences of the RNAs inside these, and that's from that it's been possible to generate evolutionary trees to indicate one slide, the relationships between organisms. Okay, so as I said, if we have the state from RNA transiting to protein, the argument for why this would have happened, given that we have an ancient system, is that RNA would not have done the job of that proteins are capable of doing quite as well. So I'll just put it as that. So what we can see is it's likely there's been a two-stage transition. One is from an RNA to RNP. Just means an RNA that's doing the reactions, kind of coated by a protein that helps to stabilize it. So we find good examples of that, where if you take a naked RNA that can do a reaction in a test tube and add proteins to it, then you get a modest improvement in their ability to perform reactions.
So what is RNP then?
RNP just means RNA plus protein, where the RNA does the reaction.
Yeah.
So here's an example then of, just to give you a sense of that direction making sense. We can see that for some sort of the average sort of turnover of an RNA, maybe about, it can carry out perhaps one reaction per minute. If you throw some protein in, you might get a doubling of that. But protein-based enzymes can perform reactions obviously hundreds of millions of times faster than any known RNAs can.
So just to make things clear, so the RNA can actually make those proteins which can give RNP?
In this case here, no, it would be just a case of a, so you can see that in just an experimental laboratory environment where you take a naked RNA and add some protein that may not have anything to do with the relation,
Okay, so
no relationship with that.
That is required to move RNA to RNP must come from some other source.
Yes, the protein will be generated somewhere. Its origin is separate from the RNA.
Okay, but somehow its presence will stabilize the reaction.
Yes. Yeah. So we may ask then why there are any catalytic RNAs left at all. And I suppose one of the reasons may be down to the size of the molecule and the rate of diffusion. So we know that roughly speaking, this is more test tube stuff, it may be trickier to say in a complex cell, but the rate of diffusion of a molecule is going to be inversely proportional to the square root of its molecular weight. So the bigger the molecule is, the slower it will diffuse. So if we think about this transition here, and why are there any of these left? Why do we find any catalytic RNAs coated with proteins when proteins can do a job better? It may be down to a difference of the size of the substrate molecule. So in the case of a large molecule, when the rate of diffusion is such that it's the slowest step in the reaction, in biochemical terms we call that an enzyme that's reached catalytic perfection, because no amount of speeding up of the reaction will speed up the chemistry of the reaction will speed up the reaction. It's waiting for the molecule to come along and bind to the active site. So the arrival of the substrate is the slowest part of the reaction. So probably we get it. In fact, if we look at known cases of RNPs, the ones that we can see, so we don't see any that are working on very small molecules. So probably this transition has gone to completion. If we look at large molecules, then we find plenty of all the RNPs have very, very large substrates.
Confused here. So on the one hand, the proteins have a much larger reaction rate, but on the other hand, they diffuse much slower because of their weight.
Well, no, so it's more like that. So just imagine the protein just standing still, or the RNP just standing still. So here's the enzyme doing its thing. If you think about it in a naive way, that because the substrate is overall smaller than the enzyme, it'll be the diffusion of the substrate to the enzyme that's going to be measurable, not the other way around. So if the enzyme is sitting still and the substrate is coming along, if it's small, it'll diffuse very quickly, which means that there can be clear gains possible at the chemical reaction step. Whereas if it's a very slow diffusion, then improvements in the chemistry of the reaction are not going to overall increase the overall reaction if you include the time it takes for the molecule to arrive and then releasing the molecule.
So the RNPs are preferable for synthesizing big molecules.
Well, it's not even that they're preferable, they're probably just not better or worse. So there's not any there may not be any reason to replace them. And that's what we were thinking some time ago when we sort of developed this type of a picture here. When we looked at sort of things that we think could be old, what we see is that the things that look old, they all seem to be involved in this process of what we call RNA processing. So we have some sort of a precursor molecule being made, a pre-RNA. Then these molecules are processed, and all of these things here are what we call these RNPs. So this is an RNP, these are RNPs, that's an RNP, and they will then process those precursors to make the mature RNAs, which will then, in fact, they all feed into the ribosome. So given that the ribosome is old, we find it in all cellular life, and this processing is required to make these pieces. We argued that perhaps all of this processing is just as old as this mechanism for making proteins. And I guess the important catch here is the title. That is that what we see when we look at this, well, I can say this bit you find in all life forms, and actually this one here you find in all life forms. These ones here are not found in all life forms, and some of them are only found in the eukaryotes, so cells with a nucleus. So these and these, for example, are only found in nucleated cells. So the possibility is that there could be later additions that have just happened along one lineage of the tree of life, or they could be older and they've been lost from the other lineages. And so we were thinking it was probably a case of loss because of differences in how the rapidity of gene expression. So on the left here is a picture, is a sort of early visualization of gene expression. So you probably can just see a very, very thin line here. That's DNA. We're looking at a single gene locus here. And what's happening is that off that DNA, a copy is being made, a working copy, a message is being made, and you can see the message here by these black pieces, and you can see these are getting longer as we go further down the molecule towards the other end of the gene. And these sort of blobby bits are actually the ribosomes jumping onto the message before it's been fully transcribed and starting to perform the translation reaction. So we see this in this is what we see in bacteria, and also turns out we see this in archaea. So these simple unicellular systems. And the upshot of that is that a bacterium recognizing the presence of a substrate, say a sugar, some source of energy, can switch on gene expression and start producing products within minutes. So you'll start to see the first fully formed proteins within 3 or 4 minutes of switching on that gene expression process. If we compare even a simple nucleated cell like a yeast, or this would also be true of a human cell, we have all these additional processing steps. So we have this processing to remove, so we make the precursor, we might have to remove some pieces, add some bells and whistles, and then export it out of this compartment into the compartment where the ribosomes are found. Then they can start translating this, and you get protein products. That takes on the order of an hour.
Yeah. So the first part actually goes inside the nucleus?
Yes, so the DNA is found in the nucleus, and the production of the message occurs in the nucleus. The message then has to be exported out of the nucleus, and then it can be translated. So those are additional steps. And if we think about that system there, it's much faster because there aren't these processing steps. So you can get proteins in minutes over here, whereas you get proteins in hours via this system. Right. Yeah. So this simplicity, and we argued then that this simplicity in prokaryotes may be an outcome of what we call R selection, which is a concept borrowed from ecology. Basically, it just means that when there are resources in abundance, you get explosive growth. So you can probably think of, I wonder if that's not working because I haven't. Anyway, what you can imagine is that in the same way as we see loads and loads of rabbits exploding in a population, the same thing will happen with an E. coli cell. So it detects that there's a sugar, switches on genes to metabolize that sugar. You use the energy then to divide. So you go from 1 cell to 2 cells, 2 cells to 4, and it grows exponentially. And this is a feature of any of organisms that are found in environments with a strong fluctuation of the nutrients. So sometimes there's a feast, sometimes there's a famine. The strategy is then to produce large amounts of offspring. In the case of eukaryotes, where there's a much slower system, perhaps it's the case that they have, on average, if you compare them to prokaryotes, they have smaller numbers of progeny. So the processing, they may, in the same way as we talked about with the rate of diffusion of the substrate to the enzyme, it may be the case that there's not a requirement for the production of proteins to be that much faster. So the synthesis, these additional steps aren't a hindrance to the reproduction, and that's simply because there's the flip side of R selection is something we call K selection, which is the idea that when nutrients are more constantly available, but only in limited amounts, you get competition for those resources rather than sort of rapid explosion of the population. So it's sort of the quality of the offspring rather than the quantity of the offspring that is important. So I'm going to now sort of play devil's advocate and suggest the flip side. So under that whole model, we're talking about in terms of selection. So we can see that there will be selection to go through this simple streamlined system in bacteria because it's fast and efficient, and if you want to make lots of progeny, that's a good strategy. In fact, in the other alternative model with the eukaryotes, the reason why we sort of argue that these things could be, these extra steps could be relics of an RNA world is that they don't seem to do anything particularly you can remove those steps, as has happened in the prokaryotes, and they seem to be RNA based. So perhaps there's just not been any selection to remove them. So rather than being useful, they're just simply not so useless that they've been removed. There's not selection against them. Okay. Of course, under this model, we would expect then that you'd have replacement of the catalytic RNAs, but more generally, there's another alternative model that has, I guess, emerged around the last 10 years that could explain this complexity, and it's called constructive neutral evolution. So the idea here, this is where I kind of haven't really finished writing that talk, so you'll have to excuse this. So the idea sort of comes straight out of population genetics, and people have been trying to explain how come eukaryote genomes like the human genome are so complex. So the human genome carries about 2% of the information in the human genome codes for the proteins, whereas it's around 90-something% in the average bacterium. Much of the material doesn't seem to do much. Half of it seems to be genes that just proliferate at the expense of the host. Why is it that eukaryote genomes are so messy and inefficient and carry so many elements that we can't really ascribe a function to? And I think the best argument that's available right now is that this is an effect of population size. So one of the other key parts of evolutionary theory, other than natural selection, is the idea that population sizes will impact evolution as a way of sorry, I'll start that again. So drift is a process that we talk about in evolutionary biology, and drift is sort of the stochastic, a stochastic non-selective process by which you may get, if you start with, for example, 2 states in a population, and by chance, if not all of the population survives, you may go from, say, equal proportions of those 2 states to one just predominating without there being any selective advantage of one over the other. So it's just sort of a random event. So when the population is small, of course, these chance events can predominate far better, such that they may actually play a stronger role than selection. So selection may remove individuals that are completely broken, but selection is going to be weakened because of this random aspect. So that means the argument then is that these complex features may evolve without selection. And I wanted to give you an example of one because I'm thinking about it at the moment. We've been doing some work on this. There is a process of, so not all the information in a genome is coded in the gene. So we might have, here's a transcript being made, and one of the types of processing we see, I'm sorry this is a complex reaction, is that sometimes that information gets changed to at the level of the working copy. So the working copy gets tampered with by a system, resulting in a slightly different working copy, and it's this different working copy, this edited version, that's used. So the information stored in the genome is not necessarily correct and it needs to be edited.
Who does the editing?
Who? Well, nobody. I guess there are enzymes that do this. So there are enzymes that will detect these sites and they will change them.
This will happen in all circumstances?
No, it's mostly actually found in organelles called. So in a eukaryote cell, you have the nucleus, which carries most of the DNA, but you also have things like mitochondria, yes, which do all the energy production. And they've evolved from bacteria, and they carry small genomes, and most of the cases of editing we see are in those or in the chloroplasts, and chloroplasts are the things that do photosynthesis in plants, and those carry DNA as well. We do find some cases of editing in the nuclear genome, but it's there are very low levels. And the key thing that I want to say about that is that what we can see is that very, very different processes of editing have emerged in different in the mitochondria and chloroplasts of different species. Probably what is happening is exactly this process of, because there's a bottleneck, so only a few mitochondria in one individual make it to the offspring of the next. You have the small effective population size, you get a bottleneck effect. So as a result of that, one of the offshoots of this drift problem is that you can get, if this is sort of the distribution of the number of mutations that are deleterious. So here are 0 deleterious mutations. So you have different individuals with different numbers of, or different proportions of these deleterious mutations within the population. Because of this bottlenecking and the chance effect, these mutations, okay, they're bad, but they're not so lethal that they kill the organism. They will, by chance, start to increase in the population. So you have the risk of losing any individuals that carry 0 mutations. So you get mutations that are carried through not because they're better, but simply because you can't reverse the mutations, and there's a bottleneck effect, meaning that drift is predominating over selection.
These are evolution of models, right?
Sorry?
This is from evolution of genetic models, right?
This is just a schematic here.
And I mean, there is also a possibility that the whole population can be wiped out.
Yeah, in principle, yeah, you either go extinct or you can start accumulating mutations. So effectively, there will be, at some point, there'll be some number of mutations that are so bad that these individuals just aren't visible in the population. Otherwise, this number could go much higher. But what will happen is that you'll get a tendency for descendants to be less fit than their ancestors, simply because of this drift effect predominating. So what we can say is that there can be, in terms of mutations, there can be deleterious mutations that are so bad that they kill the organism, in which case we never observe them in a population. If they're neutral, they're a change, but they're neither good nor bad. We can have advantageous mutations. These are going to be extremely rare. But probably most of them are going to be these, either these or these slightly deleterious ones, which can become fixed when the population is going through bottlenecks.
So when you compare a number of mutations, there can be all kinds of different mutations you're putting them all in the same basket here. Some of them might be.
Yes. Yeah, this is probably not a good saying deleterious mutations here. This is probably slightly deleterious mutations. Yeah. So there's no viability on that. It's kind of a bad graph in that respect.
Yeah.
And why can't you have reverse mutations?
So reverse mutations can occur if you have, for example, sexual reproduction. So I'll give you an example of that. Okay, just imagine we're getting mutations occur, right? So you get mutations occurring, and they can increase in population. If we have sexual reproduction, so we've got a female and a male here, we can get a process by which mutations that are bad end up in one individual, and you can get individuals carrying none. So if we focus on these two here, this one here has got so many mutations that it dies, but this one here, which has white, so if we say these are mutations, these dark things here, they're accumulating, but because we have this process of recombining, so some of the information from one parent and the other parent come together, we receive, for example, this information from this compromised individual and this from this individual. This offspring is fine. This one dies because it carries too many. So you can recombine that information to produce a individual that's free of mutations. So that's what's schematically showing here. You get some increase in mutations, but recombination will act against those.
Why can't you have the single reversal of the mutations? Because it's so unlikely. Is it so unlikely to have a reversal?
Yes, the reversal is going to be less likely because the number of sites we could potentially have a slightly damaging mutation is way higher than the number of possible reversion events. So then, if we don't have that, what we're going to get is just an increase over time of these mutations. So the number of mutations will increase. Here are slightly deleterious mutations just increasing in the population, and there's no way of purging them from the system. So the argument has been made that perhaps this editing is a way of escaping this, what we call Muller's ratchet, so it's an irreversible ratchet, because while the mutations are accumulating at the DNA level, you can fix them at the RNA level, come in and sort of change them afterwards and revert the problem that way. So how would that look? That would look something like this. Here's our DNA. This is a genetic code. there's an individual here in the next generation picks up a mutation here, and then the message is edited, and T and U, so T in DNA language and U in RNA language are identical. So this mutation is being repaired via an editing here. So even though that individual's picked up that mutation, it can be resolved.
And where does the editor know from that it has to be edited?
Right, yeah, there's lots of different systems, so that's a tricky thing. It's more or less enzymes that are naturally doing this have been co-opted, so I'll explain how that works. How it can I recognize this may be a tricky problem.
So in short, what would the answer be?
What would the answer be? What do you mean?
To his question. I mean,
I don't know. I can give you an example as to why that it may be stochastic. So I'll try and explain.
Yeah.
So, in a sense, this causes a bit of a dilemma because you sort of need. You start getting to a point where you think, oh, there needs to be a conscious editor to change this information, but it may not be the case. Instead, if you already have that activity in advance, what changes is whether or not those sites are present and can therefore be subject to editing. And then once those sites are fixed in the population, then editing becomes required. And so previously it wouldn't be required. Once repair is essential for maintenance of the genetic information, then you get locked into an editing process. So I might just, given the interest of time, skip a little bit forward. So we actually looked at this in the case of an organism called, a bacterium called Buchnera, which is a weird little thing that grows, I think if I have a picture, no? This one I have is this, sorry. So it's an aphid, this is an aphid, and inside it you have specialized cells that carry a kind of bacterium called a Buchnera. And those Buchnera are essential to the survival of the aphid. So if you feed Buchnera antibiotics, they will, the aphids antibiotics, you kill the Buchnera and therefore you kill the aphids. So neither can live without the other. And more or less what's happening is that the Buchnera, all they do is they make these essential amino acids that are not present in the diet of the insect. If we look at the genomes, it turns out that they carry, so here are some genomes from different species of Buchnera, and you can see they're evolutionarily related. So what's more or less has happened is that as the aphids have speciated into different populations, the Buchnera have come along as well. So they're inherited from mother to offspring in the same way as mitochondria are. And they've evolved from a free-living bacterium that's very close to an E. coli. So when we looked at the genomes of these, what was interesting was that some of the lineages carried, these arrows here are showing sites of mutation where if you just look at the DNA information, you don't have a functional gene that could generate a protein product. So these three over here, these individuals, for this particular gene, they're fine. But in these three lineages, they carry a mutation here. So in this case, it's actually a loss of a sequence, an A, which has meant that the information is out of frame, and the result is that you wouldn't get a protein. Turns out when we look at the RNA level, however, there's actually a stochastic process going on where the RNA polymerase, this is the enzyme that copies, makes the RNA from the DNA information. It'll hit regions, specific regions. So you can see these regions here that we're looking at. There are long stretches of A's, so there's AAA, AAA, lots of A's. It'll hit these regions and it basically gets lost along the way. You could imagine sitting there trying to copy lots of A's, you get lost. But that's more or less what the enzyme is doing. So it'll hit these regions, and for all of these genes, well, we know that there are these mutations. What we find are that here are the ones that we see from the genome in these particular cases, and here are the corrected ones. But you can see there's sort of part of a pattern where you see some that are much too short and some that are much too long for all of these loci. So it's more or less just a stochastic chance effect that some of the messages just get corrected. But it's not that it's sort of an accurate correction process, it's just that it hits this slippery region and some of the messages are then repaired. Might just skip that picture and that one. So more or less what that says to us is that, I might just cut to the chase. So how does this relate to early evolution? And then this is a case where a complex system has emerged, and it's a side effect of being a low fidelity system. So the selection is weak. You end up with sort of selection not being able to predominate, slightly deleterious mutations being picked up, and we've had this situation where there was already a propensity to edit, but you get the emergence of these tracks that where you get slippage. That will probably reduce the efficiency of the production of a protein, because some of the messages are going to be broken because of slippage. At the same time, you could get a mutation that damages that at the DNA level, so you lose one of your one part of the sequence, and the result is that you absolutely have to have the slippage in order to regenerate the correct message. So from that simple system, we end up with a complex system of expression with an additional editing step. And the point with regards to early evolution is that we can see that this is just happening under certain genetic conditions. So it may not be anything to do with being very, very old. It's simply something where the system is being perturbed such that natural selection is not operating well, and the outcome is you get these complex processes. So in the case of editing, for example, we've seen it evolving multiple times. You see it in these chloroplasts, mitochondria, and even in some RNA viruses. And it's probably the case that it's the population genetics that's dominating and switching from a sort of selection efficient system to this constructive neutral system. So I think what we're going to be I think what is happening is that we're really moving away from this idea of historical thinking to that some of these things, these processing events, are really things that have occurred as the eukaryote lineage has taken on different population genetics, such that we really can't trace these back to an early RNA world. So those principles are probably still relevant for early systems. They may not be very efficient for selection, but there's a difference between us understanding the system and us being able to have a direct historical trace. So I think that's probably where we're at the moment. I'll just end with a little comic slide there. Okay, so I'll stop there, and thanks for listening.
Thanks, Andrew. Question, please. Are we closer or further away from the understanding?
I think we know more about the systems, but I think we know, what we also know is that there may not be a stronger case for a historical trace. And I mean, that's not a bad thing. It's just a different way of doing the science now. We can start asking questions about, can we perturb the systems and recreate a complex, show the emergence of a complex process from a simple one, if we get the genetics right, which is quite exciting, I think.
Maybe it's better to say we now know better that we are actually further away.
Yeah. That kind of progress is
True, yes.
Yes, it's very easy to ask stupid questions for me. Must there be a LUCA?
No, it's probably very difficult for there even to be a LUCA. Yes, we don't really know.
Okay.
It's a nice idea. It's a convenient
Okay.
construct.
And what is the history of the ribosome? How did it develop? How did it come into being?
Right. Well,
Are there cells without ribosomes?
there are no cells without ribosomes. So we can see that there's a common core of proteins and RNAs that all cells use in making their ribosomes, and then you find some that are found only in some of the lineages, so that there are maybe some add-ons in the periphery. But all cells, if we trace that far back to our LUCA, if you like, all cells have got a fully functioning ribosome.
And roughly how many ribosomes a cell contains?
Oh, I don't know.
I mean, order of magnitude. 100, 1,000, 1 million.
Probably lots, yeah, something like that. Probably high numbers, yeah. No idea. I could look it up, but I don't know. Maybe you guys know. Do you know?
Asking me?
I'm asking the two token biologists in the audience.
It varies. I mean, the copy number in archaea, for instance, it varies a lot.
Yeah.
Bacteria, but almost the same organisms can have 4 times more.
4 times, not 100 times.
Not that I know of. I know the 4 times more, it's not 100 times.
I was quite surprised at the understanding of the self-correcting mechanism of DNA is so poorly understood. Or that is the correct word.
With this editing process, you mean? Or?
Yeah.
Yeah, I think we're starting to understand it quite well. What needs to happen is more or less a case of we've gone from the point of observing a phenomenon, and there's maybe a nice theoretical explanation for why that phenomenon has emerged many times and the commonality seems to be that it's the genetic background. What really needs to be done is testing of that to show.
But I meant, you said the stochastic mechanism for correcting.
Yeah. So what we know is that, so as I said to you, I mentioned briefly that Buchnera, this bacterium, is actually a close relative of E. coli, which is sort of the garden variety bacterium that most experimental biologists will use if they're studying bacteria. So it's sort of a standard lab organism, like a mouse or something for genetics. Or a Drosophila, yeah. So it's the bacterial equivalent of that. And we actually know from studies of the E. coli that it will slip, the polymerase in an E. coli will slip when it hits one of these regions of lots of A's. However, if you look at the genomes of the E. coli, because it's got large populations, it's got effective selection, so you just don't see any of these sites. So what's happened probably since the two, their common ancestor diverged, one stayed in an environment where population sizes were large and selection was efficient, and the ones that ended up inside the insects ended up in this bottleneck environment, so they can no longer prevent the emergence of these tracts. Because the polymerase tends to slip on them, the outcome is that you get a reduction in gene expression, efficiency, and then the emergence of corrected messages through the stochastic process. So the existence of the slippage-prone enzyme was already there in the ancestor. It's just that in one system there's effective removal of any sites that could be damaging to the process of expression, and in the other that can't be prevented.
Alexis looks skeptic. Now he looks happy again.
But if one would do stochastic editing on a text, it would never be more correct at the end than before.
Right.
It's very unlikely that it would.
No, but you have to first assume that something is going wrong stochastically,
Yeah.
and then maybe
Of
add
course.
stochastic addition to it.
So there are two things there, right? One is that we have, for every gene, there's the advantage of numbers. So you might, when you switch on a gene, you're making tens or hundreds of thousands of copies of that message. So some proportion of them may be fine, and if that's the case, they can be translated and you get a product. So there was a couple of biological slides there where I showed, not really interesting because you just see some green blobs, but we showed that this is just showing that one of the proteins that has, if we looked at the DNA, it had this frame shift that means that it shouldn't be expressed and functional. Turns out that we can see it being expressed in the embryos of these aphids. So the gene does get corrected and you do get a protein product made.
I guess maybe not up to speed, but it's also not a totally random process. So you need certain structures in the messenger RNA for the proteins to actually.
That's right. It's not random correction of a text, as it were. No.
Not any mutation can be edited like this. So only certain mutations are.
In this example, okay, this was just missing, but assume that the A would have been replaced by a C. This could be correct, but the editor would come and replace it by an A again.
No, so in that particular type of, yeah, so I showed what I would have done if I wrote this with maybe a week to spare. I would have just used one example. So there are mechanisms where you change the actual base. You might change an A to a C, or a C to an A, or a C to a U or something. And then there are editing processes where you don't change the bases at all. So all that's happening is that it's sliding along, and you're just getting you're losing count effectively. So you're getting slightly more inserted or slightly fewer. But the A to C changes a different thing again. So it's just so there are different ways in which this correction can occur on a chemical basis. And in this particular system, it's just this slippage, whereas in a plant chloroplast, it's a replacement with a different base.
That's quite something. I mean, one should maybe think. I mean, if I'm seeing a text and somebody writes the, I mean, this is one of the common mistakes. Somebody just wrote the.
Yeah,
I'm not sure that.
But there's a rock band called the. And if you would add this, then it would be wrong.
Right.
In that context, it's
Yes. Well, that effectively is probably what's happening
Sarcastic bit, perhaps.
in these guys. Is that initially you might have a, let's just say, have you got a whiteboard marker? Let's just say we had a sequence of information which was something like this, and we know that the enzyme won't make a mistake if there's unless there's around, say, let's say 9 A's. So there's a stretch here with 4 A's, then a C and 4 A's, and let's just say that there is a mutational change making that into an A. So now we have whatever that is, 10 A's. So that change may not affect the function of the product. So it could be that the information is exactly the same, but that the production of the production quality is going to be reduced because some proportion of your messages just end up being noise, because you get the slippage now occurring because that's been replaced by an A. So as a result, instead of say, like this was, I don't know, this was the average amount of material functional messages we got, you might drop that down to some lower percentage. So we get some proportion of the messages that are rubbish. So the effect is coming into it, and you get some errors that aren't errors being corrected. But then, of course, if we then have a subsequent mutation that does that, we can still produce some proportion of correct messages because of the slippage, but now the information is such that it's actually incorrect at the DNA level, so it must be corrected for that product to be made, and it may not even have any impact on the proportion of correct messages, but it means that you now absolutely need the slippage in order to produce the message.
Okay, looking outside, there's some smoke already, so we make sure it's not getting all black before we get a chance to eat. But let's thank Anthony.